Hello, and welcome to the latest installment of the National Youth Science Academy's lecture series. As always, we'll save questions until the end, but feel free to type them into the chat as we go along. We're joined today by Dr. Leo Ferreira. He's an assistant professor of microbiology and immunology and of regenerative medicine and cell biology at the Medical University of South Carolina and the Hollings Cancer Center. Without any further ado, I'll pass it over to Dr. Ferreira to start us off today.
Thank you for the kind introduction and opportunity. I'll go ahead and share my screen so you see my slides. Here they are. So it's really a treat to present today. And I was in camp back in 2019 in the summer, basically the last summer before the world went crazy. And so what I really work on, and I hope to tell you about today, is how we can design the immune system, immune cells as living drugs for a variety of disorders. So, what really motivates me is this idea that, no matter where we're from, who we are, we all experience aging and disease. That's one of the few certainties in life. And so I always wonder, since I think I was a little kid, how come that when I play soccer and destroy my knees, they regenerate back? And then someone who's old can do it less well than someone who's young. And that also holds true for fighting infectious disorders and others. And so the idea, that you have an immune system that, keeps invaders at bay, but that, gets worse over time, and maybe we can control that. That was kind of my very general idea when I was 5 years old. And now I'm in my 30s, and I still think about this stuff. And I think that's the magic of science, is that you kind of get to work on these things that you thought about as a kid. You keep playing every day. And so one very pernicious example of disease is autoimmune disease. It's an example where, the immune system, which is supposed to be your defender, it goes awry, and it's like as if the body is at war with itself. And you might have heard of psoriasis, rheumatoid arthritis, and even vitiligo and type 1 diabetes, multiple sclerosis. There's now over 80 different autoimmune disorders that, if you combine them, they affect something like 5% of people in the United States. So individually, they are rare diseases, but if you combine them all together, it's actually a pretty substantial problem. And most of them, we don't know where they come from. We don't know how to treat them, and they're painful. And most of them are controlled, but not all of them. And so this is a big problem in medicine and in science, right? So how does the immune system tell self from non-self? What does it sometimes consider self, as if it were non-self and was dangerous? So these are many questions that still today in 2023 we have. And so, you could think that if we can solve this problem, then people could live longer and happier lives. If you think about a car, no matter how old a car is, if you keep replacing the parts, the car can last pretty much forever. And so I think I'm one of those people that thinks that maybe the same could be true for people, right? If we find a way to keep regenerating, replacing, replenishing our tissues and organs, and especially in autoimmune disease where they've been destroyed by your own immune system, we could go a long way. And so, of course, it's never as simple as that. And so, as you know, if you are to take a kidney or a liver or a heart from someone else, it doesn't just get accepted by your body, right? Your immune system, your body knows it's a foreign tissue from someone else. And so then it's what's called organ rejection happens. And so again, a long time has been spent on trying to understand how can we mitigate, how can we diminish this rejection of organs? And if we could do so, then you could really open the gates for what is known as regenerative medicine, right? The idea you can regenerate lost tissues to disease with new tissues. So this is really what I work on, and I'll take you on a little, trip in time, if you will, to kind of get at the origins of this kind of research. Like when did people start thinking about, how does our immune system tell self from non-self, and how can we use this for organ transplant and autoimmune disease therapies? So the first probably hint or discovery of immune tolerance, the idea that the immune system can be unresponsive towards certain specific tissues, actually came from the Midwest, United States, in the cattle farmers. And so the idea here is that they saw empirically and serendipitously, I guess, that if you have calves that share blood flow in the placenta, so they're not, they're not real twins, they're fraternal twins, then they can accept tissue grafts from each other as adults. And this is true even if a male and a female calf. And for them, this is important because it also turns out that if you have a female calf in this situation, the calf is sterile. And so they didn't want to grow up the calf all the way to an adult cow if then it's going to be sterile. So their motivation was purely business, right? How, when should we have the cows become meat as adults or as calves? But for the immunology purpose, this was very interesting because usually the first hint that somehow the immune system can be taught to tolerate a different organism's cells, right? And so this is in contrast again with what we see in the adult, in the real world, where a kidney transplant, organ transplant in general gets rejected if it's not exactly matched and gets all the immunosuppressive drugs into the recipient. So then that was in the 40s. Now fast forward a couple decades, and now you have the work of Peter Medawar and others in England and elsewhere, where they really start playing with this and saying, okay, this is a case in nature, can I actively induce it, right? And so what they did was they found there's a critical window in development where the immune system didn't quite decide yet what's self, what's non-self. And so what they saw is that they took mouse embryos, they took, rooster embryos, and then they, at some point they took cells from another strain, right? So let's say they took cells from a black mouse into a white mouse, or the same thing for the rooster. And so what they saw is that if it is early enough, then the adult white mouse in this case would accept cells from a black mouse just fine. And the same converse for the black mouse taking cells from the white mouse, and the same for roosters and other species. And so, one step further, not only this can happen in nature, we can make it happen. But, like everything else in science and medicine, okay, we know it can happen, we can make it happen, but what's the mechanism? How can we understand it enough that now we can make it better and make it more applicable for more cases? And now we know that, there's obviously immune cells involved in this, and these are really found yet a few decades later. And so what this was now Sakaguchi in Japan, so very far away. You go from Midwest to England to Japan in this timeline. And what he saw was that there was something about, again, that early stages in the mouse, early stages after birth even. In humans, humans develop a bit earlier than mice, but in mice, even after postnatally, he saw that if you took out the thymus, this organ that we have up here, where T cells go to school, the T cells of the immune system, that, the mice had terrible autoimmune disease, gastritis, thyroiditis, and others. But if he waited a bit longer, then he was okay. And so this kind of narrowed down that there must be cells that develop in this time period in the thymus, and those cells are responsible for keeping tolerance, right? For keeping homeostasis, such that your body does not attack itself, is not at war with itself, and autoimmune disease does not develop. And so those cells then found a marker for them called CD25. So in immunology, we love CD numbers. Everything is a CD number. And so CD25 is a receptor for a cytokine, for a soluble mediator, that T cells always have at high levels on their surface. And so it was very convenient to be able to purify these cells based on this cell surface marker. And so what we saw, right, sure enough, is that if you then gave these mice these cells, you have much lower incidence of autoimmune disease. And so these cells, they first were called suppressor T cells, but they then got rebaptized, if you will, as regulatory T cells because they regulate immune responses. And that's what we're talking about a lot about today, is regulatory T cells, or Tregs for short. And so these Tregs, they develop in the thymus, and really the crux of their existence is that when T cells go to school, the immature T cells, there's kind of a range of affinity for different antigens, right? How strong do they bind to different types of protein, of pieces of proteins? And so, if a T cell is really bad at binding to these peptides, then it disappears. It's what we call death by neglect, right? There's a system where, the stronger it binds to the target, the more of a survival signal the T cell gets. And so if it's very low, the cell just dies. It's very high, the cell also dies, because then that could lead to autoimmune disease, right? Because remember, these cells are learning in our thymus, which expresses our protein. So you don't want a T cell to see our own self too well, right? So if it doesn't see it well enough, it's a useless cell. If it sees it too well, it's a dangerous cell. So both these extremes get deleted. Now you kind of have the normal range, and at the tippy top of the normal range, you have these cells that are not quite high affinity enough to be deleted, to be thrown away, but they're not normal range. And so these are what then becomes the regulatory T cells. So these are kind of the generals of the immune system, right? They keep everything in charge to make sure that nothing goes overboard. And so we now know also not just where these cells come from, but also how they work. And we're still learning, but we now know there's kind of two types of tolerance, immune tolerance: central tolerance and peripheral tolerance. And so central tolerance in the thymus, as I just described, and then peripheral tolerance is as adults. So even as we live right now, we're still teaching new tricks to our immune cells, if you will. And so at the center of both types of tolerance are the regulatory T cells, and they're characterized by this transcription factor, or a protein that binds to DNA, called FOXP3. So they have FOXP3, they have Helios. These are two transcription factors that you'll also hear a lot about. And so there's classically two main tenets of Treg suppression. One is called bystander suppression, and the other one is called infectious tolerance. So bystander suppression is a property by which, Tregs, whatever environment they're in, every immune response around them gets suppressed, gets inhibited. So even if you have a bystander cell there, it gets suppressed. Infectious tolerance is a property by which Tregs can educate, they can convince naive T cells to they themselves becoming regulatory T cells. And so this is very important because you can imagine, after a Treg cell's lifespan has expired, its effects can live on, right? Because it has effectively re-educated the environment where it was put in. And so if you look at those very generally, then I always like to try to illustrate things. So the bystander suppression is as if the Tregs can make an oasis where in this area, no overt immune responses are happening. And infectious tolerance is a bit like the Treg is teaching naive T cells so they're themselves becoming regulatory T cells. And so these are the two, classical properties of Tregs from the 1960s, 1970s. Now, of course, we're much further from that, and now we have molecular mechanisms to put to this. And there's over 12 different mechanisms by which Tregs molecularly inhibit immune responses. I just put a few of them here, and these include, Tregs make anti-inflammatory cytokines, so soluble mediators, and these include IL-10, IL-35, TGF-beta. They also modulate antigen-presenting cells. So in the immune system, the T cell does not usually get activated by a target cell. It gets activated by a professional antigen-presenting cell, which then makes it active and goes, seek and destroy, if you will, its target cell. And so Tregs act on those by modulating the APC to become less pro-inflammatory and more anti-inflammatory. Tregs also soak up cytokines that T effector cells need to survive, the biggest one being interleukin-2 or IL-2. Tregs also produce adenosine, which is a potent immunosuppressive molecule. And so by adenosine production, now the conventional T cell gets inhibited. And another mechanism is actually that Tregs sometimes, they take matters on their own hands and they just kill the antigen-presenting cell, right? So one extreme way, if you will, to terminate an immune response is to just kill the antigen-presenting cell that is causing it. And so that could be a B cell or a dendritic cell. Those are all different kinds of antigen-presenting cells. And so, all this just to say that there's many mechanisms by which Tregs work. And so that's why some of us believe that this is really going to be, if you're going to design and redesign immunology, this is a cell type that is worth spending time on because it has such a broad range of activities and they all converge on the goal of inhibiting specific immune responses. And so if we get this to work, then you can imagine, taking situations where there's an excess of immunity, which could be organ transplant rejection or autoimmune disease, such as type 1 diabetes, multiple sclerosis, and kind of, tip the scales into a situation where there's more regulation now. And so this would be if we can use Tregs as living drugs to help with these diseases. Now, a bit of a timeline, not because a history lesson, even though at this point you might think it is, but because just to kind of impress on you how fast it has been that Tregs have been from discovery to application to the clinic. It was only in the 90s that Sakaguchi found this idea that if you transfer Tregs into other mice that have disease, Now they don't get autoimmunity anymore, 90s. And then 2001 was when the human counterparts were discovered, and there was back-to-back papers from the United States and Europe and Japan. And that's 2001, right? Beginning of the century, beginning of the millennium. It only took 10 years, 2011, for people to start injecting these cells into patients. So this was first done in graft-versus-host disease, which is a terrible disease where, if you get bone marrow transplant for your cancer therapy, but the bone marrow transplant is from someone else, right? It helps fight your leukemia, your liquid cancer, but also starts reacting against your body. And so GVHD is a big side effect of bone marrow transplant for cancer patients. So that was the first, case study for Tregs. And then from that moved on to type 1 diabetes, where, you'd want to protect the pancreatic beta cells from immune attack, and then in transplant. And so there's now, at this time, 2019, when I wrote this review article, was 50 something. Now it's probably close to 100 trials with Tregs. And again, different fields. As I mentioned, there's GVHD, autoimmune disease, and then, of course, organ transplant. So these are ongoing, but none of them is FDA approved yet, which is a gold standard for therapies. And so that's because we have a way to go. So in GVHD is probably where it's most advanced, the idea of using Tregs as living drugs. You see here this data from Stanford and Orca Bio, that, if you use this regimen where instead of just giving the patient the entire bone marrow to fight its cancer, instead you sort the cells, you purify them separately, and then you first give the patient the Tregs, so the immunosuppressive cells, and the hematopoietic stem cells. These are the blood stem cells that give rise to all cell types in your blood. So that's, day 0, if you will, times 0. And then 2 days later, you give the people the conventional T cells. Those are the killer T cells. And so that delay of giving the conventional T cells 2 days after the Tregs is enough to really double the percentage of people who are free of GVHD after a year, right? So 1 year after the bone marrow transplant, you see instead of a third, now over 2/3 of people are free of GVHD. So that's in graft-versus-host disease. In organ transplant, we're not quite there yet. There's a couple studies I show here on the left. It's kind of idea that you want to wean off the immunosuppressive drugs like the tacrolimus and sirolimus and replace them with Tregs. And so there's some sort of signal you see here. The red is the regular group and C is cell therapy group, and you see a lower proportion of patients with an infection. And then you see here in the bar chart form. And so this makes sense, right? Because you can imagine that if you have an immunosuppressive drug, it's kind of a blanket treatment, right? That yes, you can now live with your transplanted kidney or liver or lung, but your immune system no longer works, right? So it's good for the organ transplant to survive, and you need that organ to survive, but then you can no longer fight infections or fight cancer. And so if you replace, at least in part, these immunosuppressive drugs with Tregs, you see an increase in people that don't have infections after their transplant. So that's a good sign. But then if you look closer, we're not quite there yet. And it's because if you now look at liver transplant, you said in general, if you try to grow Tregs from control healthy patients, healthy controls, and from liver transplant patients, you see that their Tregs from the patients, they grow less, right? So that's the orange ones, they grow less. And you see that maybe that's an easier graph to look at. So you get less cells to start with, and they expand less. And the ones that are specific for the liver, in this case, they also decrease over time specifically. And so this means that these Tregs are getting tired. And so this is actually a big problem in the field is that, you want the paradigm right now is to use autologous cells. So use your own cells to treat your disease. But if treating your disease with the standard of care right now involves taking drugs that harm the cells that you care about, then you're going to get diminishing returns, if you will, right? Because now these cells, there are less of them, they grow less, and they work less well. And so that's actually something maybe in the question session we can talk about how, we can maybe take Tregs from healthy controls and make them universal, make them compatible with everyone. And then you take those healthy Tregs into patients. And now maybe then they won't have these challenges of the cells themselves being a bit tired, if you will. So that's in transplant, that's where it's at in transplant. So yes, we can make, antigen-specific Tregs, but, they're a bit exhausted, they don't work very well. And so that's where it is right now. And then finally, in autoimmune disease, there's really, I think, the poster child, the case study, where there's a very stark difference between basic research and the clinic. So in basic research in the mouse, it's been shown that Tregs work. If you take a diabetic mouse and you transplant pancreatic islets and antigen-specific Tregs, more than half of the mice are cured, right? You see here the percentage remission. If you give these Tregs, you see it's right up there. But if you do this in humans, that's not quite the same picture. You can take 1,000 times more cells, right? So instead of 2 million, you have 2 billion cells, 2 billion Tregs per person. And, they're safe. There's no adverse effects, but they don't improve the disease. If you look at C-peptide, which is a measure for insulin, you see that, some patients keeps going down, some stay stable, but trending down. And so this means that, it's a therapy safe. So it's a good phase 1 clinical trial result, but it's not effective yet. And so one big difference between these graphs, other than mouse and human, is that on the left in mice, these are antigen-specific Tregs, right? So 100% of these 2 million Tregs are laser-focused on going to the pancreas and stopping the immune rejection there. Whereas in the human counterpart, because it is so hard to find the rare antigen-specific Tregs, these are just polyclonal. It's a bag of specificities, right? So out of this 2 billion Tregs, we actually don't know how many of them end up in the pancreas to protect from type 1 diabetes, right? To stop the progression of the disease. So that's a big problem right now. And so that made some of us think that, well, if it is so hard and laborious to isolate and expand antigen-specific regulatory T cells from humans, maybe we can circumvent this problem, this strategy, by making up our own specificity. And so that is where synthetic biology comes to our aid. So if you have a traditional, a natural, if you will, T cell, T cells get activated, they get stimulated via their T cell receptor. And this is really what gives it specificity. So the T cell receptor over there, it's binding to a peptide, so a piece of a protein mounted on, presented by a major histocompatibility complex molecule on antigen presenting cell. And so that's what we also call signal 1 for the T cell. And then T cells also need some encouragement, if you will. We call it co-stimulation. And that's given by CD28 on the T cell, another CD number, binding to CD80 and CD86 on antigen presenting cell. And so it's a combination of these two signals that make a T cell go full on blown. This T cell is now activated in response to its cognate, to its specific antigen. Now, as I said, though, it is hard to find these cells, to find the right cells. And sometimes we don't even know what to look for. Again, many autoimmune disorders, we don't know what causes them. We don't know what the disease-inducing antigen is. And so can we then, again, as I mentioned, circumvent that? And so synthetic biology, we came up, well, royal we, not me personally, with the chimeric antigen receptor, or CAR for short. So a CAR is this designer molecule, right? So designer immunology, it's a designer molecule that has an antigen recognition domain. It's antibody-based, so it can bind to anything you want, in theory, on the surface of a cell. And then you have the signaling domain. So that's how cells make decisions. And so if you see there's a CD28 domain, so just like CD28 on a natural T cell, and there's a CD3 zeta domain. So just like the TCR CD3 complex, right? Maybe I didn't emphasize that or mention it, but the T cell receptor itself does not signal, does not transduce signal. It has to associate with the CD3 complex. And so the CAR is an all-in-one shop, right? In the same single polypeptide, same single molecule, you have the antigen recognition end of the business and the signaling end of the business. And so this allows us to redirect, to reprogram the specificity of T cells to any cell surface target we want. And so this is very, very powerful. And so you probably have heard of this technique in cancer. So CD19 CAR T cells and BCMA CAR T cells, these are cancer antigens. There are now 6 FDA-approved therapies for that. And so our hope is to kind of take a page from this book and use it in regulatory T cells, so they can help underserved disorders such as autoimmune disease and also organ transplant rejection and graft versus host disease. So how can we do this? How can we, put Treg therapy in the fast lane with these CARs, right? So if you have polyclonal Tregs, I've kind of shown you in the human data, we can grow them, but then again, because so few of them actually react to the right thing, it might still lead to rejection of the organ. So then you want to have donor reactive, specific Tregs, but then these are very rare, right? You only have one cell in like a million. You need donor material as well, right? So whoever is donating the kidney or the liver or the heart, you'd also have to take other cells from this person to activate, to, selectively expand your Tregs, and then expand. And then in theory, if you transfer those, because now all of them are laser focused, it should work. In practice, however, it is hard to find these alloreactive Tregs, number one. Number two, as I showed you from the human data, they don't grow as much as we would like, and or they become, exhausted. They go bad in the process, and so they actually don't even protect as much anymore. And so that leaves us to, plan C, if you will, which is to have CAR Tregs. So in this example, using HLA-A2 as an example, or A2 for short, right? You can imagine having a donor, an organ donor, that is HLA-A2 positive, right? This is an MHC allele. There's over 3,000 MHC alleles in human, and HLA-A2 is one of them. And imagine being transplanted, given to an HLA-A2 negative recipient. Right now, we can use HLA-A2 as a marker, specific marker for the transplanted organ. So it can engineer Tregs, CAR Tregs, to recognize that HLA-A2, expand those, and then transfer those in. And now the HLA-A2 organ should be accepted. So this is what we went after. And then specifically, I mentioned organ transplants, so it could be any organ. But of course, for autoimmune disease, one that we care a lot about is the pancreas, right? The pancreas is what allows you, to control your blood sugar after you eat. And if you have type 1 diabetes, you have the bad fortune of having self-reactive, autoreactive T cells that destroy it, right? And so the idea here is that, can we stop this destruction? Can we stop this fire? And so who will be our firefighters? Will be these CAR Tregs, because again, CAR Tregs can be made specific for that transplanted pancreas. And so they can go in that area, and remember, bystander suppression, infectious tolerance, they can modulate that environment and put that fire off. So now we're, able again to control your blood glucose without having to inject insulin several times a day and measuring your blood glucose. And, in type 1 diabetes, this is already happening in people, the transplant part. This is the only curative treatment there is. Insulin controls it, but as I mentioned, you have to inject insulin every day, several times a day. But if you do a pancreas transplant, then you can have your body again take control. Now, the problem is that with any transplant, it's going to be from someone else. So your body will recognize that foreign, right? So when you inject these islets, which are the endocrine part, they're like 2% of the pancreas, they're going to now face two challenges. One of them is the same as every other organ transplant, which is allo rejection. Allo means other in Latin, and also autoimmunity, right? Because remember, this whole problem started because people, their immune system destroys their islets. So if you just put this in as is, it's like building a beautiful sandcastle by the ocean and then watch it being washed away by the waves, right? So we got to address not just the pancreatic cells, but also the immune cells. So that's where the Tregs, the CAR Tregs come in. And so what we did was we designed a CAR, a chimeric antigen receptor, to put in Tregs to specifically protect HLA-A2 positive islets. So this is a very biochemistry and immunology heavy slide, but I just want to give you the idea that, it is not it's not always easy or trivial to design these CARs. So we tried quite a few, antigen binding domains. And then in the end, what we had to do was we had to graft. So to do kind of a molecular transplant of sorts, where we took the CDRs, which are these little finger structures on an antibody, they call complementarity determining regions. So those are what kind of tells the antibody where it binds to, right? So you take those regions, those sequences, and you graft them onto another antibody binding domain. We call it SCFV, which means single chain fragment variable. And so all this means is that, it's a single chain of an antibody and it has a specific specificity, and we changed it, right? So the original specificity or receptin was for HER2, which is another cancer antigen, and we switched it to now be HLA-A2, right? And then we hope that, okay, this way we can have a CAR molecule that works. And luckily for us, it finally did. So this is what the plasmid, the construct looks like. So we have a EF1 alpha strong promoter to drive gene expression, and then we have a reporter gene, a truncated T EGF receptor. And so this allows us to keep track of the cells, right? Every time the cell makes the CAR, which is this anti-HLA-2 with a CD28 and then CD3 zeta, every time it makes this reporter gene, right? Again, this is just genetics, piece of genetics, but in the field, you have to do this so we can identify and purify your genetically modified Tregs or T cells in general. And so finally, after 2 years, we finally got this graph where we clearly show that if you introduce this CAR in Tregs, you see very nicely that the reporter gene is on and the CAR is on the surface of the cell. So we have this MYC tag. It's literally a tag. You can detect the antibody. So you see in this plot is flow cytometry, each dot is a cell. And so you see that basically every cell that has the MYC tag, so the CAR on the surface, also has the reporter protein. So that's good. So it has this rocket shape. So that was first victory. And then second victory is that the specificity is there, right? If you take target cells and there's just naked target cells, there's no activation, there's no stimulation. We looked at it based on CD71, another CD number. It is a transferrin receptor, so iron. So when T cells and Tregs get activated, they start, needing more iron to grow. And so we can look at the upregulation of that. So this is fluorescence, and the more to the right the peak is, the higher the level of the protein. And so you see that, if you have naked cells, nothing going on. But if you have HLA-A2 expressing cells, now you see activation. And so this peak number 1 and peak number 5 are positive controls, right? If you activate T cells via their TCR CD3, and CD28, then of course they also get activated. That's our positive control. But the real experiment is peak number 2, where peak number 2 is up, but then peak number 6 is not up. So it's specific. So we made anti-HLA-A2 CAR Tregs. The CAR is on the surface, and it stimulates the Tregs in response to the presence of HLA-A2. Now, of course, in this business, it is as important what it binds to is what it doesn't bind to, right? You want, designer immunology, you want to be specific. And so you don't want a CAR that is HLA-A2 and 3 and 4 and 5. You want to be only 2. So luckily for us, HLA-A2 was the first MHC molecule to be crystallized, actually by my PhD advisor, Jack Strominger, back in the 1980s. And so it's really been very well studied. So we pretty much know the entire map of the surface of the HLA-A2 and its applets. And an applet is just a name for an immunogenic surface, a surface of a protein that can be recognized by the immune system. And so you see there's all these different applets. These are amino acid one-letter codes, right? 41T, 90D, 80I, and so on. So these are all surfaces that poke out of the HLA-A2. And so we want to know, what does our anti-HLA-A2 CAR bind to? Do we have to worry about cross-reactivity with our HLAs? Because again, you don't want the CAR Tregs to get distracted, right? If the CAR Tregs were to now traffic and be activated by not just the transplanted islets, but also by other organs in the recipient, then your efficiency goes down and we're back to ground zero. So you want to make sure these CAR Tregs are specific for HLA-A2. And so we use binding beads, we use cells, and so I'm just showing the cell data. It's the more exciting part, where if you take human pancreatic islets from different people, and you take some HLA alleles that share some of these applets, some of these immunogenic surfaces, you see that the HLA-A2 CAR that we made recognizes, gets activated by only in the presence of HLA-A2. So you see A2, you see a shift, you see a 71 peak. But if you have HLA-24, HLA-11, 31, which they share some of the applets with HLA-A2, right? For example, one of these ones here, it's going to be also here, but it's not there, and in the other alleles. So no activation, right? So this means that we got a good product, right? It is specific for HLA-A2. Okay, so we have that. Now, how do we take this one step closer to actually be used in people, right? So far, we've been using lentivirus, which is great. Many trials use this, but there's some dangers associated with it. One of them has to do with the fact that it's random integration. So imagine it integrates into a cancer gene. Right now, these cells can become cancerous. And so this is actually still today a concern. Another alternative would be to use adeno-associated virus, AAV for short. And so AAV is great and also more trials with it, but it does not integrate at all. And so the problem there is that it gets diluted out, right? So the more your Tregs grow, if they grow 10 times, 100 times, by the time your final product, cellular product is done, there's no CAR left. And so that's not interesting to us. We need to have genomic integration. But ideally, we want it to be precise. And so that's where CRISPR comes in, CRISPR-Cas9, right? Because we can now have a Cas9 protein, right? So it's an enzyme, right? It cleaves DNA and direct it to the guide RNA that is specific for the TCR locus. So the T cell receptor locus in the genome of the T cell, you can direct the Cas9 there and make a cut, a double strand break. And when a cell gets a double strand break, it immediately wants to repair it. And so you're going to fool the cell, trick the cell into repairing it with your gene of interest. So you are effectively inserting the CAR, the HLA-A2 CAR, into the endogenous TCR locus. So this way, it's no longer a random integration. It's only one integration per cell, because in T cells, even though they have two TCR alleles, one of them is silenced. So one copy per cell, and it's going to be expressed, is going to be made at the right amount, because T cells know how to express T cell receptor, right? So now they're going to use that knowledge to express the CAR. And this is again possible thanks to CRISPR-Cas9. And I'll give you a slide of a parenthesis. Just like Tregs, it took them 10 years to go from being discovered to going to people. The CRISPR-Cas9 field is actually even faster, because it took people even less years than that to go into people. It was only in 2013 that CRISPR-Cas9 was described in human cells, and then same year it was described in mice. So you can make genetic mouse models much quicker with CRISPR. And this was, Harvard, Broad, MIT, and this was right around when I was there. Between 2011 and 2016, I was in Boston at Harvard doing my PhD, and so we wondered, well, cell lines, mice, that's great, but how about human blood cells? And we immunologists, we care first and foremost about blood and about primary cells, so they can actually inject into people. And so in 2014, actually, we were the first to publish CRISPR in primary human blood cells. And so this is 2014. I think it was November 2014. Now, next year, the following year, in China, they added human embryos. And in 2017, also in the United States, at Oregon. So, in a few years, we went from showing in a cell line in the dish to making human embryos with edits. Now, these embryos were not, they were terminated, right? They did not lead to a pregnancy. This is like just all in vitro proof of principle. But then, of course, as you know, in 2018, human babies were born whose embryos had been CRISPR edited. So in 5 years, in 5 years between 13 and 18, we went from doing CRISPR-Cas9 human cells to human babies. And then, of course, there's a moratorium. I'm not encouraging anyone to make CRISPR human babies, but it did happen. And so as such, I guess it gets me the timeline. And then, back to the normal world, if you will, there's now in vivo clinical trials where instead of taking cells out, CRISPR modifying them, putting them back in, you can just put in the CRISPR directly. And this is since 2020. And now this year, 2023, might actually be the year that the first FDA-approved therapy, CRISPR therapy, is going to happen. So CRISPR Therapeutics is actually co-founded by one of my PhD advisors, and I helped a little bit at the beginning. That's why I bolded these two events. This is my self-serving bolding. But so CRISPR Therapeutics and Vertex, they now have a therapy that has been submitted to FDA for approval. So this could be the year that CRISPR therapies get approved by the FDA. And so that would be 10 years that it takes to go from cell line to therapy. And again, that's the parenthesis, if you will, for CRISPR. But so back to our project, how can we use CRISPR in this specific project? Well, we can use it to insert our HLA-A2 CAR, right, A2, C28, C3 zeta, into the track, into the TCR locus. And then that's kind of the timeline there. You get people's blood, you get Tregs, you activate them, you CRISPR them, and then you add the CAR as an AAV, because remember, the AAV does not integrate, so it doesn't stick around. And then you make your cells. And of course, you want to compare your A2 CAR Tregs with your unedited and resting controls. And so, first things first, did editing work? And the answer is yes. You said here, you see now these Tregs are HLA-2 CAR positive, and they're TCR negative. So TCR based on CD3. So they are in this lower quadrant here. They are HLA-2 negative, of course, otherwise they activate themselves. The other important thing is that, are they still Tregs? Right? Do they become some other cell type? And the answer is they stay Tregs. They remain high on FOXP3 and Helios expression. And finally, the specificity also is maintained, right? So if you have HLA-2 positive cells, they get activated. Now, what does this mean in terms of function, right? That's really what we care about. So I'll show you one in vitro assay. It's called in vitro suppression assay, and it's pretty much a titration. So in this axis is how much cells suppress. And remember, Tregs inhibit the proliferation, the cell division of conventional T cells. And so what you see here is that, the more Tregs you add, so 1:1 is 1 Treg for every T effector cell versus 1:10 is 1 Treg for every 10 T cells. You see that the more Tregs you add, so from right to left, the less the T cells proliferated. So this demonstrates that in the presence of HLA-A2, HLA-2 CAR Tregs suppress the proliferation of T cells. So that's promising. But now we got to do in vivo, right, in a mouse in this case. And so we have two models there. One model is GVHD. And so here we have 8 groups, but again, it's a complicated slide. But the main idea here is that if you have HLA-A2 either given by the white blood cells from the donor, right? So I have HLA-2 positive donor there. If you take these cells, inject the mice, and then, as you can imagine, if you put human white blood cells into a mouse, the mouse suffers GVHD, right? Because these human cells, they see the mouse as foreign. And so these gray groups here, which only get PBMC, which stands for peripheral blood mononuclear cell, they're gonna suffer the most. And then we hope to alleviate that suffering with HLA-2 CAR Tregs. And so there we have either in regular immunodeficient mice, these are mice that don't have an immune system, so they allow us to study human immune cells in vivo in an animal model, and then you have HLA-2 transgenic NSG mice. So these mice, they're immunodeficient, and they express HLA-2 themselves. This is really cool because this better mimics what happens in the GVHD patient, right? In a GVHD patient, the bone marrow transplant is attacking the tissues of the recipient. So we have these two mouse models, that's these two sets of columns, and then on the horizontal axis, if you will, you have donors that are either HLA-2 negative or HLA-2 positive. And so to show GVHD, and so combining all these data, you can see that every group that received HLA-2 CAR Tregs in the presence of HLA-2, either in the mouse host or the injected PBMC or both, the CAR Tregs protect. This is survival here. And you see that if you don't give HLA-2 CAR Tregs, all mice have perished in 2.5 months. Whereas if you give CAR Tregs, 40% of them are still alive. And then there's a group of mice that they keep going for almost 3 months. Roughly 10% of the mice, they survive for 3 months with this treatment. That's survival, right? That's a big picture. We also try to get some more insight into the mechanism. And so we bled the mice and look at the blood. You see that if you have PBMC alone, you have very nice engraftment, presence of human CD4 and human CD8 T cells. So the CD4 are the helper T cells, CD8 are the cytotoxic T cells. But in the presence of Tregs and HLA-A2, so not this first group, right? Because group 1, 2, if you look there, is group 1, 2, there's no HLA-A2, neither from the mouse nor from the PBMC. So it doesn't work, the Tregs don't work, but the other 3 groups, you see much decreased numbers of human T cells in the blood, and obviously much better survival. And if you actually are to gate to select on those HLA-2 negative cells, which are the Tregs, they remain FOXP3 positive. And so this is encouraging and kind of matches what you see in people in the clinic, which is you inject these Tregs and they go into these hostile environments, but weeks later, they still remain Tregs. They did not convert into effector cells. And this is a very important point because the last thing you want when you work with living drugs is that your living drug changes on you, right? So you infused a patient with an anti-inflammatory regulatory T cell. You don't want those cells to now become pro-inflammatory effector cells. And so it is very important to track over time if your Tregs remain FOXP3 positive, which is a marker for them, being anti-inflammatory. So as a GVHD, we also have an islet model, right? So for type 1 diabetes, so there, we can make the mice diabetic by destroying their endogenous beta cells, and then we can take a human islet transplant to the kidney capsule. So it's a very convenient spot to put human islets because it's accessible and highly vascularized. So the islets there can sense blood glucose and immediately secrete insulin. And also, luckily for us, human insulin works on mouse tissue. So these mice, once they get the islets, they're now cured of diabetes. And then you wonder, okay, if I now infuse CAR Tregs intravenously, which in people is not on the arm vein, but in the mice is either retroorbital, so in the eye, or in the vein. In this case, we're doing tail vein here. Do they go to the right place? Because remember, that's the whole point, is that we're making laser-focused Tregs to go to the islet. And we see that is the case. If you look over time here, if you have polyclonal Tregs, what was done in the clinic for people before, they don't always stick around very much. This is a heat map for luciferase, so firefly luciferase, so you can label and find the Tregs inside a living mice. All these mice are alive. You look at the cells inside them in real time. So they go away. But if you have an HLA-2 CAR, now they survive and they go to the right place. So the spleen, right, of the mouse, and it's on the left side, so it starts there. It's a blood cell, of course, it has to go to the spleen, but then over time, you see trafficking, it sees moving over to the graft. And if you look 2 weeks later, 5 out of 6 mice still have the CAR Tregs nicely concentrated in the islets. And I don't show here, but importantly, these mice remain normal glycemic. Their glucose levels remain low, which means that the CAR Tregs were not nefarious, did not harm the islets. So the CAR Tregs laser focus on going to the islets, and they were not detrimental, right? So they didn't hurt. So that's where we are right now. Now we're testing, can the HLA-2 CAR Tregs stop an ongoing immune response against the islets, right? So with the mouse models, you kind of always try to get closer and closer to what happens in people. And so part of our ongoing research is to better model human type 1 diabetes in humanized mice. So that's where we are now. And so this is in mice, and then I'll show you a couple work from other people as well. So it's not all my work. So in monkeys also being done. So rather quickly, but just to show you that, the non-human primates, much closer model to humans than mice are, if for nothing else, because they're much bigger, to kind of get a better idea of how many cells you need for a person. And so you see this article here that came out last year, and I actually got to meet the first author also last year in person. And so you see they sort the Tregs just like we do for human blood, and then they engineer them also again like we do for human. And then they do dress in islet transplant. So here they chose the bone marrow as a site for islet transplant. And so you see that only the islets that have in this case, HLA-BW6, so similar to HLA-A2, survive. The other ones don't. And they also show that CAR Tregs nicely, they traffic and they stay in the site. So if they give 2 Treg injections here, and there's those 2 peaks there after the arrows being injections, and you kind of see here, FOXP3, Treg marker versus CAR, you see that population there is only present right in the site that has islets. If you don't have islets there, there's no Tregs. So again, laser focus to go to the place where they are needed. So that's non-human primates. And so now, of course, it's led to an explosion of companies, of biotech companies working on this. There are several companies, including one of them, Sonoma, that was founded by my postdoctoral advisor. But others also in California, these 2, and in England, in London, and also one used to be in France and now moved to the US. And so all these companies, or most of them, that's their cell type of choice, the FOXP3 positive Treg. And so I'm showing you this, just this table to kind of get an impression of like now in 2023, right? This is all happening as we speak. There's all these different indications that we really, the field as a whole is making this a reality. Right, we are going to make CAR Tregs a FDA-approved therapy for patients with autoimmune disease and organ transplant rejection. So that's the companies, and then one of them already has kind of a white paper, or it's a paper, but it's kind of a schematic only of how they're going to go about it. So on the left, you have your schematic again, where you take cells from someone, Tregs, engineer them with HLA-2 CAR, the one I told you about, expand them and infuse them into the patient. And this patient is, in this case, a liver transplant. And so in clinical trials, you always want to try different doses. It's called a dose escalation because, we want to kind of balance efficacy with safety. So if you give more cells maybe you get more efficacy, but maybe some toxicity also starts happening. So that's why they have different cohorts with low, medium, and high dose. And then this is ongoing again. This is happening as we speak. There's no results yet. Now, in a few minutes, I just want to tell you like what else is going on. Like I've shown you all this, like we know what Tregs are, or for the most part, we can purify them, we can genetically modify them, and we have ongoing trials in people for organ transplant autoimmune disease. So what else, what else has been worked on? What do we have to improve on? And so just rather quickly, I'll go through some, slides. So one thing is, can we grow them more, right? So I showed you that some patients actually don't meet the dose needed because the Tregs just don't grow enough. So can we maximize that? And so one pathway that we looked at is the tumor necrosis factor pathway. And this is because TNF-alpha has two receptors it can bind to, TNFR1, TNFR2. And while TNFR1 is bad news because there's a death domain, so the cells die, TNFR2 is good news because it's a survival signal. And Tregs, turns out, and they have a lot of TNFR2. And it's actually, in your body, that's actually a mechanism by which Tregs suppress effector T cell function is by soaking up the TNF based on their high expression of TNFR2 compared to T effector cells. And so we wondered, well, what happens if we add this cytokine? Because TNF-alpha, as the name kind of indicates, tumor necrosis factor, is seen as an inflammatory cytokine. Another inflammatory cytokine is IL-6. So these you expect to be bad for Tregs, but actually it turns out they are excellent for Tregs. If you have purified human Tregs and you throw in TNF-alpha and IL-6, their growth explodes. You can see here on this graph, when you add both IL-6 and TNF-alpha, in the presence of IL-2, I should mention Tregs need their IL-2 at all times. So always, you see there's no condition without IL-2. But so if you have that, combination of three cytokines, you see that they grow way more. And this is even more pronounced here. You now get, 1,000-fold. You get 1,000 times more cells than you started with. If you have this optimized growth expansion protocol of IL-2, IL-6, TNF-alpha, and C28 superagonist. So it really binds C28, the co-stimulatory domain, and really activates it strongly. And so this is, just to show you that we're still learning what Tregs like, what Tregs need, and how can we make more of them to inject into people. And of course, I had showed the functional outcome, which is we make more cells, but it's not like each cell is worse than the other ones. They are equally as good. You see in vitro that they suppress the same way. And then in vivo in the mouse, this is the same GVHD model where you see if you have the PNC alone, the mice all perish. But if you have Tregs, either with the traditional way in blue or new way in red, you see that the survival is extended to the same extent. And so this is one exciting outcome because sometimes you just need more of them. We never have enough Tregs, it seems. The other thing also is the heterogeneity of Tregs, right? So as you know, now with the advent of single cell, cells that we thought were similar are not so similar, and so Tregs do not escape this trend. And so if you are to take Tregs from the blood of healthy donors and GVHD, for example, and you do, single cell sequencing, you now see that they actually subset. And importantly, again, I'm very kind of function-focused person. It makes a functional difference, right? If you take some of these subsets here, the red and the purple subset, so the red and purple, they're over here, so P7 and P8, which are, of course, the more rare ones. They always say that the best essences are sold in small bottles, and I guess Tregs are no exception, right? So the rarer Treg subsets also happen to be the best at suppressing. And so again, you could imagine in the future, you can further subdivide Tregs into better subsets to be even more, effective when given to people. And then, these are from blood. The same is true from tissues. So actually, blood is very important, but a lot of action in the immune system happens in the tissue. That's where a lot of immune cells live. And so Tregs are no exception. And actually what's been interesting over the past, more than a decade now, is that Tregs in different tissues, they look different. And the best example is the adipose tissue, fat. Fat resident Tregs express PPR gamma, which is a fat cell transcription factor. So to me, this is really crazy how Tregs, they kind of, by osmosis, they learn from their environment. And so if you take a Treg from fat, some of his genes are going to be the same as fat cells. And the same would be true for muscle Tregs, for intestinal Tregs, and so on. And so it's also an emerging field for diseases of different tissues, pancreas, central nervous system, thyroid. Do we want tissue-specific Tregs in addition to the specificity? Do we also want the tissue profile to be the same? And so this is our data still unpublished in the works and others, we working at this. So with single cell sequencing and now taking CD4 cells, so effector cells and regulatory cells, and activating them either via the CAR or the endogenous T cell receptor, and we see differences. And so the hope of all this is that we can learn, genes that can serve as markers to select more suppressive cells, or genes that we want to ablate, to delete because they're causing trouble. And if we delete those genes, those cells will be more potent. So this is really very much in the work. Like, how can we improve? So making more Tregs, making more adequate Tregs. Those are the two big things, right? So it is, I hope I convinced you that, we're on our way to making regulatory T cells as living drugs for a variety of diseases. And so really have to, reach what I like to call the 4 S's, They have to be specific, right? That's the whole point of an immune cell. They have to suppress. That's the function of a Treg. They have to survive. So again, Tregs are a bit more fragile than conventional T cells are. So this is a big point. They have to survive, they have to grow well, and they have to be stable. So Tregs, if given extremely inflammatory environments, they can become effector cells and lose their function. So you want to be sure that their function is stable. So those are the 4 S's. And so there's a few questions here that I'm not going to go through this. We kind of talked with them over the talk. And so in the last couple of minutes, I'm going to show you that, there's a world besides Tregs. Because remember, at the beginning, I promised you designer immunology. So I don't want you to think that it's only Tregs are part of the designer immunology movement. And so a couple examples. One of them is from the aging world. So my very first slide. So a lot of aging seems to be caused by accumulation of senescent cells. These cells are zombie cells. They don't do anything good anymore, but they refuse to die. And so what was found recently, a couple years ago, is that if you make CAR T cells that kill senescent cells, now you see an improvement. So they have several models. One of them is liver fibrosis. And so you see if you give these CAR T cells, you now see much less of a fatty liver, and you see less of senescent cells, which are stained here by beta-gal in blue. And so again, CAR T cells is not just to kill cancer or the CAR Tregs for type 1 diabetes. They can use for something as general as, helping to cleanse your body of unwanted senescent cells. And then, the other thing I want to say is that they don't have to be, taken out, modified, and put back in. You can actually make the CAR T cells inside the body. So there's, this idea, I think, actually with the COVID-19 vaccine, mRNA vaccine, this idea of using lipid nanoparticles in T cells inside the blood. And in this case, this CAR recognizes FAP, fibroblast activated protein. And so why is this interesting? Because activated fibroblasts are a big problem in fibrosis. So we have a heart injury. The fibrotic heart is because of fibroblasts. And so what they showed in these in vivo generated CAR T cells that they improve. If you induce heart injury in mice with hypertension, that's the angiotensin 2 there, you see that in the presence of the CAR Tregs, they look, almost as good as mice without injury. So you see that red bar versus the gray bar. And so the heart you see here on the rightmost, it's much smaller and less inflamed based on this marker here. So, again, this is very early on. It's just coming straight from the lab. But I just want to kind of show you that there's multiple immune cell types and multiple ways of engineering immune cells that can really expand what design immunology can do for us. And so that's my last slide, to show you that basically, I like to look at the immune system as a group of cells screaming to be repurposed as living drugs for a variety of diseases to fight autoimmune disease, cancer, and aging. And so that's really what we've been working on for quite a while and will continue to. And so with that, I want to say, I had the pleasure to be at camp back in 2019. And big thank you actually to Mohammed Mosawa Raji. He's my friend and is one, who got me invited to go. He himself is an alumnus. He's from Bolivia. And he went as a high school student and then as an instructor together with me in 2019. And then this is my lab and our funding sources, the usual NIH, also the Medical University of South Carolina, where my laboratory is, and other sources like the Human Allergy Research Network for diabetes, lab members, collaborations. And then, with that, my lab is in Charleston. So if you're ever around, or let me know. I'll be happy to chat. Happy to take any questions. Thank you.
Great lecture as always. Thank you very much for joining us. We have a few questions from the chat that I will read out to you, and looking forward to your feedback. So the first one, it comes from us when you're talking about the type 1 diabetes slide. And the question is, why do the alloreactive Tregs not get rejected?
Well, so the alloreactive Tregs are from the recipient, right? So this is an autologous cell therapy where you have a type 1 diabetes patient and you take their Tregs, and those are the ones that are alloreactive. So the alloreactive Tregs will not be rejected because they are part of the same immune system that is causing the problem. Now, the follow-up to that is, well, can we use someone else's Tregs, and would those be rejected? And then the answer is yes. So that's why I think I briefly mentioned, but you might want to make them universal. So there is now a whole line of work I didn't mention here, but I have some work on that as well, where you can engineer immune molecules on the surface of cells to make them no longer be recognized by the immune system. So they become invisible, if you will. And so then if they become invisible to the immune system, now they can cross the immune barrier, and you can have the centralized cell bank that can be given to multiple patients instead of having to take from every person their cells and put them back in. Yeah.
And our second question, how would you propose to circumvent CAR Treg exhaustion from chronic activation/stimulation, such as through transient rest to boost functionality and reinvigorate the CAR Tregs?
Oh, that's an excellent question. We actually don't know yet. So T cell exhaustion is much better defined in conventional T cells, right? So in the cancer field, there's all these markers for exhausted T cells. They're tired. They see antigen a lot. As it's mentioned, they become activated over and over and over again, and they no longer function. Tregs, it's not clear yet either a similar problem. So Tregs, going back to the very beginning of where Tregs come from, Tregs are, biased towards self-antigens, our own antigens, and they see them over and over again. So Tregs are more robust, if you will, to this, exhaustion from excessive activation. That said, data from patients, from the liver transplant patients, right, it showed that over time, the antigen-specific Tregs were slightly decreasing. So there is, it seems now that we're doing this in people, that there is indeed a risk of the Tregs, needing some reinvigoration. So I think that there's a few things here to be worked on. One thing is, can we optimize the CAR receptor itself, right? So the CAR is the way it sees antigen is via an antibody-based domain. So antibodies, what do they have? They have different affinities, different avidities. And so you can imagine playing with this to find what's a sweet spot, right? Where the CAR Treg sees the target, but doesn't see it so strongly, then it gets tired of it, right? And then the other side of it is in the intracellular domain. You can imagine adding, signaling is a bit like playing with Legos. You take these domains from different molecules and you put them all, stack them all together. And so also work ongoing is, can we improve on the signaling portion of the CAR, specifically for Tregs? So, to maintain their function and their stability for longer. So that's an excellent question. I hope to have a real answer than what I just gave you in maybe a few years. Yeah.
Awesome. And to kind of follow up that question from the same person, they follow up with Weber et al. Found that SRC inhibition with datatinib helped reinvigorate exhausted CAR T cells and facilitating epigenetic remodeling. Could this be applied in the case of CAR Tregs?
It could be. So I'll tell you my very honest answer, which is, so Tregs are a type of T cell. And so you'd expect that, they're a type of T cell. So whatever you find in conventional CAR T cells for cancer can be applied, can be, inferred, if you will, for Tregs. Not true. If you take, for the classic example is, if you take 28 CD3 zeta and a 41BB CD3 zeta, these are the two CAR signaling architectures approved by the FDA for CD19 CAR T cells. And they both worked fine. There's differences. CD28 zeta is a bit more explosive, 41BB zeta is a bit more long-term, but they both work. Now take those CARs for Tregs and it's like day and night. 28 zeta, the Tregs, the CAR Tregs get activated, they suppress. 4-1BB zeta, they don't. And so that's just a very easy example, if you will, of how something that works or goes a certain way in conventional T cells does not translate well to Tregs. And so the answer, even as it sounds unfortunate, is that we just have to try it out. We have basically what I've learned over the past, I think 7 or 8 years now, is that whatever you see in conventional T cells, you try it out in Tregs, and it might work, might not work. You have to really do the experiment. Yeah.
Great answer. And to kind of wrap us up, Randy H. just wanted to say, amazing lecture. Thank you for your diligence in examining the published SARS-CoV-2 T-cell paper. Love your work. Love your letter to the editor. Great work.
Thank you. Yeah, this is, 2020 was a crazy year. And so, yeah, I did help a paper get retracted because, it was just very bad work. And I think it was a weird time where everything got blown out of proportion. So it scared a lot of people because they claimed they found that SARS-CoV-2 could infect T cells. Who does that? HIV does. And so people then was like, okay, is this a new pandemic that we're all going to get immunodeficient from it? And the answer is clearly no, right? I mean, SARS-CoV-2 is not yet to be found to infect T cells. And so that's why, I took the time off my day, I guess, to make sure that paper went down. And it did. It took 3 or 4 months, but the paper got retracted. No longer in the literature. Yeah.
Well, awesome. Thank you very much for joining us today. Awesome lecture. We had over 120 people join us throughout. So wow. I'm sure that everybody was able to pull away at least something. But it seems like there were some very active and engaged participants in the chat. So again, thank you for joining us. And we hope to see you all for the next alumni lecture series.
Thank you. My pleasure. Thanks for hosting.